Professor Forney is with us. Professor Forney, are you with us?
Yes, I am.
Okay, great. So, good morning. My name is Corine Jackman, and today I will be discussing the work that I've done for my dissertation, which uses a microfluidic co-cultivation technology platform to further elucidate and investigate bacterial interactions that are specifically from the human vaginal microbiome. There are approximately 10 times more bacterial cells than human cells in your body. The Human Microbiome Project set forth to understand the relationship between microbial composition and human health risks. In their study, they had over 200 people participate and donate body samples that were over 18 different sites. For example, one of the sites was the nasal passage, the oral cavity, skin, the gastrointestinal tract, commonly referred to as the gut, and also the urogenital tract, which for this purpose we will refer to as the vaginal microbiome. In 2011, there was a study that used independent sequencing in order to determine the most dominant bacterial species that dominated each woman of reproductive age. Each of the points in this tetrahedron represents the most dominant bacteria. There are 5 different vertices that are shown by this tetrahedron. The 4 Lactobacillus species are Lactobacillus crispatus, Lactobacillus gasseri, Lactobacillus iners, and Lactobacillus jensenii. Lactobacillus are lactic acid producing bacteria, and most of which are associated with promoting health. However, there is a 5th group. This 5th group is known as the diversity group and is composed of bacteria such as Gardnerella vaginalis, Mobiluncus, Antipobium, and many more. People who are dominated by diversity group, for example in this tetrahedron, are considered to be healthy. But what is interesting is that the diversity group, or the bacteria in the diversity group, are actually associated with bacterial vaginosis. What happens is, when there is an increase of bacteria that are associated with the diversity group, sometimes there can be an imbalance, and sometimes the number of bacteria from the diversity group increase so much and such that the Lactobacillus species decreases. When this happens, there can be imbalance, which can lead to dysbiosis. One example of dysbiosis is bacterial vaginosis. Bacterial vaginosis, or BV, is a polymicrobial syndrome, and it happens to be the leading cause of women's health care visits in the United States of women of reproductive age. What happens is when women have bacterial vaginosis, they have an increased chance of acquiring HIV and other sexually transmitted infections such as chlamydia, gonorrhea, and when these are untreated, they can progress into pelvic inflammatory disease, which can lead to infertility in women. One question that you may be asking is, if all of the women happen to be healthy in this study, but there's quite a few of them who are dominated or have high prevalences of diverse bacteria that are associated with BV, how can these women still be healthy and mostly be colonized by species from the diversity group? And how do the different bacteria interact with each other over time? Well, I'm glad you asked. And this is a question that we have had, and in order to help understand these interactions, we have employed a microfluidic co-cultivation technology platform. In simpler words, these are basically microdroplets that are being generated using a microfluidic device. The device that we're using, you can tell by the geometry, is a flow focusing geometry, which we will talk about later. Each of these microdroplets are about the size of the circumference of a strand of hair. And they are on the nanoliter to picoliter scale. Each of the microdroplets can be used or referred to as a miniature bioreactor. And one of the advantages to using microdroplets is that it enables a more high throughput experimentation. And also because they are so small, they have wonderful confining properties. that allows us to confine small microbial subsets and take advantage of any kinds of small amounts of fluid. So in our case, vaginal fluid. So now that we've been introduced to microdroplets, I'd like to talk more about how we can actually use them to cultivate bacteria. So first, we have 3 different microdroplets with different kinds of cells. The different cells are represented by the shapes and the different colors. This is called encapsulation or confinement. And then we cultivate them. The conditions for cultivation are dependent on the species that you would like to cultivate. For our work, we use the anaerobic conditions because the vagina is known to be a hypoxic environment, which means limited oxygen. So as you can see, there is one microdroplet that did have growth, and we would call this cultivation. And there are 2 droplets that did not. Now after this, you can use all of the microdroplets, or you can isolate a single microdroplet and have some kind of analysis where you can use something like quantitative polymerase chain reaction, qPCR, or you can enumerate the cells onto agar medium, or you can do 16S rRNA gene sequencing, whichever you choose, or if they're fluorescent, you can do FACS, fluorescent automated cell screening, or sorting. So now that we have learned how to encapsulate and characterize these cells, I'd like to let you know that there are a lot of different interactions that can be observed using microdroplets. For instance, we can see mutualism. Mutualism is when 2 different species are unable to grow on their own, as shown here. So these show, and then they are able to grow when they're in co-culture. So we see this cell, this is a red rectangle, and this blue circle. They're both different types of species. And when they grow on their own, called singleton droplets, they're not able to grow. However, when they are together, they are able to grow. This is called mutualism, and was demonstrated by someone in our lab called Ji-Hung Park. And she published this study in 2011. And here we can see 2 different E. coli auxotrophs in a single microdroplet, which are shown here. One is labeled with YFP and another with M. cherry. They are fluorescent. And when they are co-cultured, you see that they grew after 18 hours. However, when you have a singleton droplet with each of them, we see that there was no growth after 18 hours. And this demonstrates mutualism. There is another type of interaction known as amensalism. Amensalism is when both species, as shown here, are able to grow on their own. However, when they are co-cultivated, we see that only one species is able to grow. This is because one of the species has an inhibiting or killing effect on the other, due based to molecules that are produced and inhibit the other. But you have another type of interaction called competition. And I think of this as resource competition, where both species are able to grow on their own, but when they are co-cultivated, one is able to get to the nutrients before the other, or faster than the other, and you can see that one outcompetes the other and has higher fitness in that environment. Now that we have learned about the different interactions that can be observed and detected in microdroplets, I'd like to share with you the different aims and platforms that we have used in order to further elucidate microbial interactions in the vaginal microbiome using a microfluidic platform. First, it's important to show that microdroplets can indeed be used to understand and investigate interactions. And so we have extended a technological pipeline in order to recapitulate an interaction that was already reported. Secondly, we wanted to investigate some of the pairwise interactions between Lactobacillus species, most of which are associated with promoting health in the vagina. And we did this using standardized and synthetic laboratory media, which is commonly used to cultivate Lactobacillus species. And third, we understand that people act differently in different environments, and the same can be understood for bacteria. Bacteria behave differently depending on their environment. And if we use more of a natural simulated milieu or a vaginal environment, like a vaginal fluid, we want to eventually culture the bacteria and also further work, study interactions between them. And so with these 3 different aims, we would like to develop and demonstrate the high throughput nature that microdroplets have in order to elucidate some of these interactions in the human vaginal microbiome, and also exploit the confining properties of microdroplets to show how we can work with small volumes. And eventually this technique can also be used for many other ecosystems, not just the human vaginal microbiome. Aim 1. So this work was basically inspired, or as part of it was a report that we looked at was from Atasi in 2006, where she was able to show, he or she was able to show Lactobacillus gasseri cells and Lactobacillus gasseri byproducts and their effect on Gardnerella vaginalis. The y-axis shows the colony-forming units of Gardnerella vaginalis, and the x-axis is time. And we can see that when the cells are in co-culture with Gardnerella vaginalis, they are able to survive. But when we mix the byproducts of Gardnerella vaginalis, within 1 hour, viability has dropped by about 7 log. And this would show a killing effect that this specific species, Lactobacillus gasseri, has on Gardnerella vaginalis. Some of these mechanisms for exclusion are considered to be lactic acid, which is produced by the Lactobacillus species. And there are other mechanisms as well, putative mechanisms. But because we were able to see how Lactobacillus and its ability to produce lactic acid indeed does inhibit the ability for Gardnerella vaginalis to grow, we wanted to further demonstrate this, use this platform and demonstrate it using microdroplets. And then in a second pair, we wanted to further demonstrate it using another species. And it's important to note that the species we chose is Gardnerella vaginalis, which has been associated with bacterial vaginosis, and another bacterium that has been associated with aerobic vaginosis, a less publicized but polymicrobial syndrome. So the first step in order to apply our approach or develop our approach was to first choose our model system, which we did. It's Lactobacillus jenseniae and Gardnerella. And then we confine the microdroplets. As you can see here, we have 2 droplets in the microdroplet as they're being generated, then we have 3, and in some cases we have 4. This happens in real life, and it's because of what is called the Poisson distribution, which has a range of different number of particles or cells per droplet. Then after we have encapsulated the bacteria, then we cultivate them. As you can see, the monoculture here on the first and the last microdroplet have grown. But we see that there's some kind of an interaction here, and if you remember, this demonstration is just one of those interactions, the mensalism. And then there were 2 different assays that we had in order to analyze the cell content. And our main goal here was to enumerate or compare the growth when a species was in co-culture with the other, and compare the growth when it was in monoculture by itself. So the first assay was by pooling microdroplets and using quantitative polymerase chain reaction, qPCR. So what we did was we pooled the droplets together and then destabilized them and used this technique, which does not involve cell viability but only the DNA content. And then we used a spacing device in order to isolate single microdroplets, destabilize each of those single microdroplets, and enumerate the colony-forming units from each of the microdroplets in order to compare and contrast the number of cells in monoculture and co-culture. So these results are from a flask culture. Before actually doing the microdroplet experiment, we wanted to show that we can show a similar interaction in flasks. So on the left-hand side is Gardnerella vaginalis, and on the y-axis is the fold change of colony-forming units. And the black bar represents Gardnerella in monoculture by itself, and then Gardnerella is in co-culture with L. jenseniae. The 4 asterisks here represent a significant difference between Gardnerella and vaginalis, such that Gardnerella is inhibited by Lactobacillus jenseniae. In the case for Lactobacillus jenseniae, we see that there is NS, which is no significance between the 2 different conditions. And so we say that using flask culture, we saw that Gardnerella vaginalis was inhibited in co-culture, but Lactobacillus jenseniae was not. So now we are moving into the microfluidic aspect of the project, of this aim. So first, I'd like to show you the actual device that we use. The device can be held in the palm of your hand, and it's comparable to the size of a penny, as you can see. And the actual material is made from PDMS, polydimethylsiloxane. And it's commonly used because of its advantageous properties, which include being chemically inert, being non-toxic, being readily producible, and optically clear. And when the microdroplets are generated, we have the media, which includes the bacteria, and that is diluted to whatever dilution you would like in order to have a certain lambda or average number of cells per droplet. Then we have oil that comes in, and as they shear together, flow through the orifice, they generate confined and monodispersed microdroplets. So afterward, in panels C through H, we can see 6 different microdroplets. On the left-hand side are the microdroplets at time 0, and on the right-hand side are the microdroplets at time 12. And what you can see is that there are more cells after 12 hours than there are at 0, which would indicate growth. And you can also see that the cells are more rod-shaped and they tend to agglomerate for panels C and D, which panel C is Lactobacillus jensenii. When it grows, as you can see in panel D, it tends to just have more of the same. And you can see that this kind of is comparable to that. And when you look at to that, meaning panel F, which is in co-culture, and when you look at panel G, this is Gardnerella vaginalis. They're very, very small cells. They're coccus or circular shaped. And when they grow, these cells are planktonic. No, qualitatively, when looking at the microdroplets, you may be able to see that Lactobacillus jensenii is going to grow and basically take up most of the space in the microdroplet. And this would indicate that Lactobacillus jensenii would have an inhibiting effect on Gardnerella vaginalis. Now, before going into the results from spacing, I'd like to show you what we use. So this was developed by Stephen, and as you can see, looking at the inside of the microfluidic device, you can see that when we release pressure from this quick valve, droplets flow from the microchannel. And you might have just seen that 2 of them actually escaped at that point, but really the goal is to have 1 microdroplet escape at a time. And also the device that we use is set up here where the microdroplets flow through this tube and go into a microcentrifuge tube. And to make sure that we are actually isolating a single droplet, we did some assays. And we used fluorescein microdroplets in order so that we can visually see them. And we kept working with and developing and optimizing the system so that instead of collecting 4 microdroplets, which are seen here, we can eventually have a higher probability of isolating 1 microdroplet. This is 1 microdroplet in a micro well, and this what you see here is a reflection, and that's also a reflection. So now we're looking at the results from pooling the microdroplets and doing qPCR, conducting qPCR on the pooled droplets on the left-hand side. On the left-hand side, we have the estimated number of cells. They are estimated because we are working with the DNA content and correlating this number back to the number of cells based on a standard curve that's not shown here. And we have Gardnerella vaginalis here, and the black bar is in monoculture, and the white bar is in co-culture. And as you can see, Gardnerella vaginalis has grown more when it is in monoculture than in co-culture. When looking at Lactobacillus jensenii, there are 2 asterisks here, which was different from the flask results. But we must remember that qPCR is different from that in flask, and I will explain why. Lactobacillus jensenii in monoculture has grown more, but we see that there was a difference between monoculture and co-culture. And we see this, and we've repeated it over and over. We've seen the same result over and over with these experiments. And the reason why we think this happens is that we see this interaction in both cases with Gardnerella and with L. jensenii is for a few reasons, and that is because we are using qPCR, which does not rely on viability of cells, only on DNA content after growth. And also because we are pooling microdroplets. When microdroplets grow, as you remember, like the Poisson distribution changes the number of cells that are initially in the droplet. But when we combine 1,000 microdroplets, as we did in this case, approximately 1,000, we increase the signal-to-noise ratio. And in that way, we're able to cancel out any noise. And also, we just know that qPCR is a very highly sensitive technique. In the case of enumerating the cells from individual microdroplets, on the y-axis, we see the colony-forming units per microdroplet. And then we see Gardnerella vaginalis on the left. The blue circles indicate the number of colony-forming units that were detected from each microdroplet. And there were 10 microdroplets for each condition. As you can see, there is quite a bit of variation, but the cells are present. We have 1 all the way up to the order of magnitude of 1,000. And when we counted the colonies that were for Gardnerella vaginalis in co-culture, It's important to note that the same microdroplets that were enumerated, that were counted and processed or analyzed for co-culture, we split the contents in half such that the number of colonies that were present here were the colonies were the cells from the same microdroplet that was for Lactobacillus jensenii in co-culture. So as you see, Lactobacillus jensenii in co-culture had quite a few colony-forming units that came from the microdroplets, but all of the cells, this indicates all of the cells were dead in Gardnerella vaginis, or did not grow on colonies on the agar medium. So this would suggest that there was an inhibiting effect. However, because we have such a large range here of colony-forming units using individual microdroplets, this was unable to give us any significance in terms of these results. And the reason might be because Gardnerella vaginalis is known to be a fastidious bacterium, and also looking at the phenotypic differences between each cell. There are differences, just like there's differences in each of us, but we're all of the same species. There's also differences between each cell. And also the Poisson distribution, meaning that there are differences in the inherent differences in the number of cells that are initially in colonies, in droplets. And for Lactobacillus jensenii, we did not detect a difference for either condition. So in order to further demonstrate the general applicability of our system, we have employed a second model system, which works with which includes Lactobacillus jensenii and Enterococcus faecalis. Enterococcus faecalis is associated with many infections in hospitals and has quite a few virulence factors, and is associated with aerobic vaginitis, along with other species, but we have chosen Enterococcus. It is also much more robust than Gardnerella vaginalis and grows very well. There was a study that was conducted in 1996 where the adherence of Enterococcus faecalis was shown to decrease when it was with Lactobacillus reuteri, and when it was not, it was able to adhere better. So first we conducted a flask experiment, and in the y-axis we show the fold change of colony-forming units, and on the x-axis we see Enterococcus faecalis, and right away you see a big difference between monoculture and co-culture, in that Lactobacillus jensenii, when it was in co-culture, it inhibited the growth of Enterococcus, but we did not see a difference for L. jensenii. This would show amensalism. And then, in terms of microdroplets, we can see that the microdroplets were able to support the growth of the species. So when you see Lactobacillus jensenii and you see the cells that are inside the microdroplet, you can see how much they've grown. We've also grown them in a different media, and because we were growing different species. So it was MRS media, which is ideal for Lactobacillus species. So Lactobacillus jensenii has agglomerated and more rod-shaped. And this same morphology and growth seems to be present in this microdroplet. However, Enterococcus faecalis is different. Its morphology is more heterogeneous, and we can see that they don't tend to clump in microdroplets as much. And so qualitatively, this would indicate that Lactobacillus jensenii was inhibiting Enterococcus faecalis. So using our platform assay, when we isolated microdroplets, we were in fact able to indicate that Lactobacillus jensenii did inhibit Enterococcus to some level of significance. So here we see much less variation, but we do see Enterococcus was able to grow in microdroplets, and we see that Enterococcus in co-culture was able to grow, but there were differences, and this was statistically significant. When looking at Lactobacillus jensenii from each of the individual microdroplets, there was no significant result that was seen significant difference between the two different conditions. And so, in summary, we have demonstrated that Lactobacillus jensenii indeed has inhibited Gardnerella vaginalis when flask culture, and then also with using qPCR. And then we were able to detect amensalism in our second model system using our individual droplet platform. So now that we have demonstrated that microdroplets can be used in order to demonstrate different interactions in microdroplets and between bacteria, we wanted to investigate potential interactions between Lactobacillus species. using synthetic laboratory media. There are 4 different Lactobacillus species, but 2 of them happen to be the most dominant species in the vaginal microbiome. They are Lactobacillus crispatus, which is shown in red and is associated with health, and Lactobacillus iners, which is shown in orange. And it's debatable whether it is associated with health, but there are some characteristics that have associated it with possibly being a pathogen or possibly being a commensal organism. And so this graph represents the temporal fluctuations of Lactobacillus crispatus and Lactobacillus iners, and if you look closely, there are some other bacteria that were present. But subject 28, which was the healthy woman of reproductive age in the study, was mostly colonized by L. crispatus, but it changed every 6 weeks and so on as time went on. And this type of study is called a longitudinal study. Also, the y-axis is the type of bacteria, the relative abundance of each of the species. And so when looking at Lactobacillus iners and Lactobacillus crispatus, there was work done in 2016 by a person named with the last name France. And what they did was they predicted and they compared the accessory genes, which are genes that are specific for that species or that strain, and then they compared the core genomes for both of them. And so what we found is that, well, when looking at this pathway, we can see that the black arrow represents shared genes or shared pathway between Lactobacillus iners and Lactobacillus crispatus. So for example, both species are able to metabolize or uptake glucose, and then both species are able to produce L-lactate. But in the case of the other D-lactate, only L. crispatus is able to produce this. There are also other nutrients too. So both species are able to uptake maltose. However, fructose, which can also be found in the vagina, is mostly taken up by Lactobacillus crispatus, and there were not genes to show that L. iners was able to do this. And so because there are differences in terms of niche or in terms of ability to compete for different resources, we wanted to further understand how these bacteria compete. And this was a prediction. These were predicted, but we wanted to empirically demonstrate any kind of resource competition or other types of competition that may exist between these 2 most dominant species. So another resource that is potentially introduced into the vagina, which we will talk more about, is in the form of menses, and can be, in menses you have red blood cells which contain hemoglobin, which contain iron. So we wanted to demonstrate the possible influence or investigate the possible influence that iron can have on Lactobacillus crispatus and Lactobacillus iners. And secondly, we wanted to examine or investigate the potential effects of competition between Lactobacillus crispatus and Lactobacillus iners, and also a second model system, which we will talk about later, briefly. So there are differences between Lactobacillus crispatus and Lactobacillus iners. Lactobacillus is associated with health due to its production of lactic acid, T and L, but Lactobacillus and other mechanisms, but Lactobacillus iners actually persists at a higher pH, a pH of 7. And it also persists during bacterial vaginosis when other species, Lactobacillus species, tend not to. And it also can persist during metronidazole treatment, which is what is used, antibiotic that's used to, for temporarily get rid of BV. And also the genome size and GC content are different between L. iners and Lactobacillus crispatus. For example, L. iners has a genome size of 1.24, which is within the range of a parasite or a symbiont. However, Gardnerella Lactobacillus crispatus has a genome size of 1.83 and 2.46 megabase pairs, which is higher. Lactobacillus iners has a GC content of 32.7, which is lower, and then crispatus, which has a GC content of 37. A species or just a lower GC content is indicative of having higher activity in what is known as horizontal gene transfer, which is incorporating genes from its neighbors or other bacterial cells. Having a smaller genome or that of a parasite can suggest that Lactobacillus inners may not have enough genes to perhaps survive in more of a not turbulent, but an environment that is dynamic and disturbed. And this was actually predicted by someone else, but we have found almost the opposite, where Lactobacillus inners is able to persist in more dynamic environments, such as after coitus and during menses and even BV treatment and metronidazole. And so we know that in ecology, we say that no two species can have the same niche and still coexist, but we see that there's this temporal fluctuation of the bacteria. So it seems like there may be some kind of a apparent or fundamental niche overlap where they are able to metabolize similar resources. So I wanted to investigate that empirically. So before showing results, I wanted to more in depth show the differences or show the changing microbial composition that can occur within a woman. This graph, this chart, represents one subject and how her vaginal microbiome changes over time. We start off with diverse group, and over time it moves to a Lactobacillus crispatus, and then it moves closer to Lactobacillus inners microbiome. And then it starts to return back to Lactobacillus crispatus. And then after about week 11 or 12, we see that it has went back to a diverse microbiota or microbial composition. So this graph looks pretty cool, but how is it that this happens? We don't really understand. We don't really know, per se, what causes these changes or what are the mechanisms by which these changes come about. However, we can say that there is a mechanism that has been predicted, and it's called conditional differentiation, which occurs when there happens to be resource competition. And so conditional differentiation is when the availability of a certain factor, whether it's abiotic or biotic, is present. And when it is present or not present, it will affect the fitness and competitive ability of that species. So some of the carbon sources that can be shared are glucose and maltose, but there are others that are shown that cannot be shared between both species. Carbon sources are very important for these bacteria. Also, iron. The host, when I say host, I mean human, the host has a tight restriction on iron, and this could be because iron is known to increase the ability for pathogens to grow, because pathogens tend to need iron in order to grow. However, we know that when the time of the month comes, there is iron that is available. Lactobacillus crispatus has been sequenced, and what we have seen is that they have ABC transporters, which include siderophores that can actually acquire iron. Lactobacillus inners has also been shown to thrive in some cases when serum that contains iron or when some types of blood have been added to media where they previously were not able to grow. And so what we wanted to do was to investigate whether or not the presence of resource competition or interference competition could be detected using synthetic laboratory media. So with resource competition, you have two different species, and they're competing for this, in this case, the simplified schematic. They're both competing for the same molecule of glucose. However, there's another type of competition, which is called interference competition. And this is when one species is actively aggressive towards the other. In this case, perhaps Lactobacillus may be secreting lactic acid, which would inhibit Gardnerella vaginalis, for example, which we saw in AIM-1. So first what we wanted to do was measure the effect of iron on Lactobacillus species, so in this case Lactobacillus inners and Lactobacillus crispatus. And then we wanted to determine or investigate potential resource competition and also interference competition between Lactobacillus crispatus and Lactobacillus inners. The results that we're showing today are only for Lactobacillus inners, but at the end I will tell you the results that we saw for Lactobacillus gasseri. Lactobacillus gasseri was chosen not because it was one of the most dominant species, but because it is one of the four most occurrent or prevalent of Lactobacillus species in the vaginal microbiome, but it was shown to not transition to Lactobacillus crispatus, which was interesting. So our hypotheses, we had two. First was that the availability of iron, of free iron, would stimulate the growth of Lactobacillus inners. The test for this was to measure the growth of Lactobacillus inners and Lactobacillus crispatus after they have been supplemented with iron sulfate and with an iron chelator called 2,2-bipyridine. Second, for the results, the addition of iron did not stimulate growth. However, when we did add the iron chelator, which reduced availability of free iron, we saw that growth was reduced. For our second hypothesis, we wanted to investigate the potential for competition between Lactobacillus and synthetic laboratory media. And we conducted this by doing continuous serial dilution co-culture experiments and also experiments with spent media in synthetic laboratory media. And the results were that neither resource competition nor interference competition was detected using synthetic laboratory media. So these are methods that were used to do the first experiment that we talked about, where iron was added in the form of iron sulfate, iron chelator was added in the form of 2,2-bipyridine. We use MNC broth, which supports the growth of Lactobacillus inners and L. crispatus, but already includes some amount of iron. And it cannot be, if it is removed, then L. inners won't be able to grow because serum needs to be added to the media. Also, we incubated them or cultivated them in a 96-well microplate. We incubated them anaerobically at 37 degrees Celsius for 24 hours, and we used 3 different biological replicates. This is a picture that shows the reaction where you have iron, and you have 2,2-bipyridine, and it forms a complex around iron so that it potentially is harder to be able to acquire. And we had a range of 2,2-bipyridine between 0 to 1.5 millimolars, and then 0 to 100. And in our second experiment, we increased the number of conditions between 0 and 1. And just to remind you, MNC is the control where we did not add iron or an iron chelator, but there was still some amounts of iron that were present, which was why we added the chelator to decrease the availability of free iron. These are the methods that show how we were able to conduct our supplemented or media in terms of looking at how L. inners growth and L. crispatus spent media is influenced. And so we grew the cells and removed them, and then we added iron, and we also added MNC powder in order to introduce nutrients back into the medium. We had 2 different biological replicates of L. crispatus to observe any differences between strain or between the replicates. And then we had 2 different technical replicates of each condition and 2 technical replicates of each control. Now we're seeing the results. So on the left, there's Lactobacillus inners, and on the right is L. crispatus. MNC is the control where no iron or where chelator was added for both graphs. And as you can see, there was hardly any growth here. And the y-axis is the change after 24 hours of optical density, or OD. And so at 1.5 millimolars and 1.1, you see that the growth was greatly reduced, but this was not shown for Lactobacillus crispatus. When we look at the very right, we see that there was no growth when 100 millimolar of iron was added to both conditions. Now on the left, this is because of the 2,2-bipyridine, which did not have the same effect. And this could be for a few reasons. Perhaps the 2,2-bipyridine, it did form complexes with iron, making it more difficult for Lactobacillus inners to acquire iron. However, because L. crispatus did not have the same effect, perhaps it has siderophores that can more efficiently acquire this iron, or maybe Lactobacillus crispatus just does not need or require iron. Also, a potential mechanism for the inability for these species of L. inners and L. crispatus to grow, or they did not grow, might have been because of a Haber-Weiss reaction. But that is speculated. So in order to look at differences between concentrations of 2,2-bipyridine and its effect on L. inners growth, we grew Lactobacillus inners up to 30 hours, and we saw that as you decrease the concentration of 2,2-bipyridine in increments, you also have increased amount of growth, which is an inverse relationship. But you can see that there was indeed a difference, and where this is 1.5, 1, 0.7, and 0.4, we see that 2,2-bipyridal did have an effect. Also, we wanted to see whether when Lactobacillus inners was grown, whether L inners would be able to grow when we added glucose or added iron, and we saw that it was not. It only grew in MNC, which is likely because there were more nutrients that were needed in order to grow these species. In interest of time, I'm going to just say that this is a co-culture experiment, a continuous co-culture experiment where serial dilutions took place over time. It was cultured anaerobically, and we used Lactobacillus crispatus and L inners. As you can see, there are 3 different conditions. We have a comparable, which is the initial ratio between L inners and crispatus. We have more amounts of Lactobacillus crispatus and less amounts of Lactobacillus inners for the initial concentration. The vertical purple lines here show the dilutions that took place, and then so the co-culture was shown to grow collectively, so this has both species, and then it would decrease, and then it would increase. So the decrease shows the dilution, and the increase shows that they were able to grow. The pH was also monitored. We wanted it to be at 7, and this was shown by the turquoise dot, and when it decreased, it was brought back up to 7 at different points. And the composition is also shown where L. crispatus is in the orange and Lactobacillus inners is in green. So if there was resource competition, we would have seen that there was an overall trend where one species would have gone a certain direction, but we don't really see that here, no matter what the different conditions were initially. And so we say that we did not detect a difference or resource competition between these species. And the initial seed ratio between cells can be reflected also at time 0 when looking at the relative abundance of species. We also wanted to look at the interference competition between both species, and in order to do this, we used a similar technique as before, where we grew cells, removed cells, and then we added back MNC powder. Now, the reason we do this is because if we're testing for interference competition, that would mean that there's molecules that would inhibit the ability for the species to grow. But by adding nutrients, perhaps this inhibiting molecule would still have a negative effect on species. However, we did not see that. When looking at Lactobacillus inners in L. inner spent media and Lactobacillus inners in L. crispatus spent media, we can see that the PBS, which is a saline solution, Lactobacillus inners did not grow, but when we used Lactobacillus crispatus spent medium, they also did not tend to grow as much, or they basically declined. But when we add the nutrients back, they were able to grow. So this would show that there was not really an inhibiting molecule that affected growth or viability of L inners. In the case of Lactobacillus crispatus, first we see L. crispatus was able to grow more, or that it didn't decrease as much as L. inners, because L. inners is very fastidious. But first, when we did not add any nutrients, there was a great decrease. But when we did add nutrients back, it was able to still survive. So we did not detect interference competition. And so in summary, we were able to show that reduced availability of 2,2-bipyridal did increase 2,2-bipyridal, reduce the growth of Lactobacillus inners, but not of L. crispatus, and that other limiting nutrients there were other limiting nutrients that existed besides glucose and iron, and that no interference competition was detected in using synthetic media between L. crispatus and L. inners, and we also saw the same for our second model system with L. gastroi and L. crispatus. So now we've used the synthetic laboratory media, but the thing about using synthetic laboratory media is that the components inside of it do not tend to be well defined, and they tend to be a mixture of different molecules or compounds that do not exist in the vaginal microbiome or in the vagina. And so there were some efforts to make a medium simulating vaginal fluid, where in this case the glucose competition of that and the glucose concentration of that of vaginal fluid and that of medium simulating vaginal fluid are similar for different compounds as well. However, for example, there are over 600 more proteins that are present in vaginal fluid that are not present in this simulating simulated vaginal fluid. And so what we wanted to do was to find a way to develop and then implement the use of human vaginal fluid, cervical vaginal fluid, which in this case I will call CVF, and use that to grow Lactobacillus species and eventually in the future be used to detect interactions. So first we wanted to stimulate the vaginal milieu and then exploit the capability of microdroplets that can confine small volumes of fluid, specifically because a woman can donate anywhere between 10 microliters to 600 microliters, but in order to do one droplet experiment, we can use 30 microliters for droplets, and droplets work at the nanoliter scale or picoliter. So in order to investigate or further start this process, we first collected samples, vaginal samples from women. They were self-collected, and we had in total 49 samples that were collected. Then we used a rayon swab, and this rayon swab, it was not cotton, so we wanted to avoid sequencing a cotton gene, so we used rayon. This swab cup is a menstrual cup that is sold over the counter and is used to collect menstrual fluid, but for this case, we collected CVF or CVM. Afterward, we submitted our samples to the Microbial Community Analysis Board, where they did DNA extraction, qPCR, DNA sequencing, and then after the results came back, we processed them using a platform or a bioinformatic kind of software known as Mother. And afterward, we were able to begin implementing our CVF to culture Lactobacillus species. So first, we did submit vaginal swabs instead of CVF, which we used, and literature did show that they were pretty compatible, but we wanted to demonstrate this. So VS stands for vaginal swab, and CVS is cervical vaginal fluid. And so we see that the microbial composition is similar for subject 48. It was similar for subject 49, similar for subject 50, and similar between subject 51 and 51. And so this demonstrates that we can indeed, or verifies that we can use vaginal sample swabs instead of CVF for sequencing. And this represents the microbial composition of all of the samples that we have received. Most of them are Lactobacillus crispatus, as you can see, and because of this, we chose Lactobacillus crispatus as the species in order to pool our vaginal fluid, because most women who were dominated by who had high prevalences of L. crispatus, this would help us increase our volume. In order to normalize for the volume between different women, because different women can donate and have volumes of 10 microliters to 600 microliters, we added the same volume for each person. So we allotted amounts of 40 microliters from each woman in order to have a pool. And this pool was composed of women who were mostly colonized by Lactobacillus crispatus. Then we cultured the bacteria in microdroplets. So Lactobacillus inners, known to be quite fastidious, hard to grow. We cultured this and we used two different pH values for L inners. It tends to prefer pH 7, but the natural environment tends to be 4. L. crispatus, we grew this at pH 4, which is the preferred pH for L. crispatus and preferred for L. gasseri. And then we had empty droplets to make sure there was no contamination. We grew them anaerobically and in microdroplets. So this shows Lactobacillus crispatus at 0 hours and 24 hours with fluorescence overlay and at bright field, and we saw that they did not grow, which was very surprising because it was L. crispatus dominated vaginal fluid. And we think reasons for this may have been because there was not enough nutrients in the vaginal fluid for the CVF for them to grow. And this also could have been that perhaps there are some compounds from the host or signaling molecules that are produced from that host constitutively that would allow them to grow. But because we have removed them from their natural environment, they no longer would be able to receive these molecules, potential molecules. But in the case of Lactobacillus inners, a very fastidious, hard-to-grow organism that does not even grow in MRS, which is used to grow most of the Lactobacillus, it was actually discovered in 1999, way after Lactobacillus, we found that it did grow. And Lactobacillus crispatus dominated CVF. But it grew only at pH 7 and not so much at pH 4. And pH 7 was chosen for L. inners because it's more preferred. And so this was very surprising. But when we actually pooled them together, we see that the pH tended to have a specific effect on L. inners, where the pH 4 is shown here, and pH 4 of L. inners is in blue. And so we can see initially at time 0 that there was a drop. for L learners at pH 4. But while L crispatus and Gasseri were at pH 4, they had a viability that was much higher than that of L learners. And after 12 hours, L learners at pH 4, the colonies were no longer detectable. But L learners at pH 7 almost reached what was higher than 1 times 10 to the 9 colony-forming cells per unit. Colony-forming units per colony-forming units. And then for Lactobacillus crispatus, which is purple, they were able to survive, and then L. gasseri decreased within 24 hours. So in summary, we were able to demonstrate or show that Lactobacillus inners was able to grow in LC-dominated CVF. And we also developed media to simulate the vaginal milieu using pooled LC-dominated CVS. And then we adopted our microfluidic platform to confine small volumes, in this case CVF. In conclusion, we have demonstrated the effectiveness of microdroplets using our model system between L. jensenia and gardnerella and Lactobacillus jensenia and Enterococcus faecalis. We have also detected no interaction between the Lactobacillus species, L. inners and L. crispatus and L. crispatus and L. gasseri, and we showed that the addition of 2,2-bipyridal did reduce the growth of L. inners, but not of L. crispatus. And also, we were able to develop and implement media that mimics the vaginal milieu, and also showed that when adopting microdroplets, we were able to confine CVF in order to culture bacterium. Future work involves a use of this fluorescent species, or fluorescent strain of L. crispatus. Fluorescence increases the capability where we can now use fluorescent automated cell sorting to do more high throughput automatic screens. And also, it was not mentioned in this work, but where the location of sampling also will affect microbial composition. Lactobacillus inners is actually the most widely detected bacterium, but was not the most widely detected here, and that might have been because of ethnicity or demographics. And so changing location can also select for different species, and if we were to grow bacteria in Lactobacillus inners dominated fluid, perhaps L. crispatus would grow. And in order to do interactions, you would need both species to grow. So with that, I would like to have a sincere thank you for everyone who has helped me along this journey trying to answer these questions. I'd like to thank Professor Lynn. You have been a tremendous help for me, and you have helped me so much in terms of growing and in your support. This was truly a transformative experience for me, and I will remember you forever. And Professor Forney, I want to thank you for your support. Although you are at the University of Idaho, you have definitely helped me to grow, especially in terms of the way I think and writing and presenting also. Professor Foxman, I want to thank you so much because there are so many other directions that we could have gone in, but with your experience and with your expertise, you have been able to guide us, especially when working with human samples. And Professor Gulari, you have also been a tremendous support for me. I want to thank you for all of your advice that you've given me throughout these 6 years. You have been very helpful. And Dean Solomon, I want to also thank you. Some of your questions and your contributions have helped me to be able to think differently and more of high level in order to think about what direction we're taking. All of my lab members, our work is different. No one else works on the vaginal microbiome, but you guys have been very attentive and you've asked questions that have helped me along the way so much. So I want to thank you all for your individual contributions and your attentiveness. And some members are not here, but I also thank them. This University of Michigan is really big, but we have been able to find different people who have helped us. Our CBS underwent mass spectroscopy analysis, and that was due to the help of the chemistry department with Professor Robert Kennedy and his student Matt Sorensen. I learned how to culture anaerobic bacteria in the Tom Smith lab and took Vincent Young's class on learning about microbial interactions, so I want to thank them for their help and support. Jason Bell, there's something that was not included in this dissertation, but I actually went to the hospital to start collecting samples there. We filled out an entire IRB through the medical school, and he opened up his team to us to me while I was there, so that I worked with them, and they helped me to collect even more samples for a completely different study. And also, the University of Idaho, Professor Lynn allowed me to go to the Forney lab, who was on the call, he's the committee member, in order to learn how to culture Lactobacillus species and just learn more about their environment. And what I found and learned, the people I met were truly instrumental for my journey. And I remember these people, and they have helped me so much, even with sending me strains and just advice along the way. And Kanetta is still in the lab, and I talk to her now. So they're all very helpful. So with that, I'd like to thank my funding sources and thank everyone for being here. And thank you so much.
Okay, so the next part of this process is to have all of you raise questions. And I'm sure Corine will try to answer them as well as she can. From the back? Yes.
When you culture the bacteria in the droplets, do they stay on the droplet, or do you put them on some kind of like substrate where they adhere and then the media dries out, or how does that work?
Yes, so very good question. I've never been asked that question before. So when you generate the microdroplets, the bacteria are in the droplet as well, but they're not applied to any kind of substrate. The droplets are there and they kind of form their structure around each other. But no, they're able to stay in the droplet. What we find is that when we lay them out on a surface, like in a C-chip, we find that over time the cells begin to settle to the bottom, which are great for imaging. But they are always able to stay, at least for the cells that I work with. There was a former member in the lab who was trying to grow, I think it was trichomonas, and it has more fibers, and that would stick out of the droplet. It was unable to stay in. But for our purpose, they were able to stay inside of the droplet. Thank you.
More questions? We have another microphone. You want to ask something?
I'm Cara Evans. I'm a friend of Corine's. I have a question for you about your sampling. So when you were working with the collecting the samples in the hospital, were there certain criteria that the women had to have as far as to make sure to normalize your experiment? So were you looking at all women, or there women that were a certain age? Were there women that were that had bacterial vaginosis in the past? So were there any criteria that you were looking for, or that any criteria you were trying to exclude?
Yes, absolutely. So not only in the hospital, but also when we collected samples here, there was definitely criteria that actually, I think, excluded quite a number of people. First, the women had to be of reproductive age, which for our purpose was between ages 21, it could be lower, but it was 21 to 45. And it was open to students, faculty, and staff, but students were the people who donated. And they had to be healthy, meaning that they couldn't have had I don't want to go into detail, but they had to be healthy. They initially could not be on birth control because hormonal birth control is known to affect the vaginal microbiota. But Jason Bell has work that suggests that it does not have an effect. So I think that's something that could use some further discussion and just thought or some time to talk and discuss about. Let's see. Yes, there was a lot of criteria. I think I have a slide on it, but yes, there was quite a bit of criteria.
Thank you, Corine, for this wonderful presentation. So based on the conclusions that you have now, and as you move forward in your career, what are some future directions that you're thinking about as you're moving forward?
Yeah, so that is a great question as well. So I have an opportunity to do more research, and I'm thinking of moving away from vaginal microbiome only to expand more into other microbiomes like the lung. I initially was thinking the gut, but there's more opportunities for lung. But in terms of vaginal microbiome, I was thinking there are some questions that interest me particularly. For example, people have seen that there are differences in terms of diversity among ethnic groups. And also in terms of using microdroplets, which I think is a tremendous tool that can be used. I think that doing more experiments to collect different samples, and if we collect the ones that are enable growth, because there can be some technicality with that because we want to make sure the results are reproducible. But at least seeing if we can actually kind of develop a more, I guess, a pipeline that is able to grow more than one species and then look at interactions between them. I mean, that's very close to what I've done, so I don't know. We'd have to talk about what I would be able to do. But also, like, it would be great. A lot of people in the lab they have fluorescence in their cells, so that wasn't, unfortunately, we weren't able to really do high throughput screening in terms of like using FACS. But if we were able to use this strain, there's a lot more experiments we could have done. For example, also what we were planning to do was co-culture Lactobacillus crispatus with like natural microbial subsets from women with BV and women without, just to see what effects that would have had. But yes, and I do plan to think more about the future too. No question.
Great job, especially for those of us who are used to what you were doing. On the statistical side, then, can you let us know like what the level of significance was? Was it 0.05, and what types of analysis tools that you use on it? You can check these on distribution and also. So I don't know if it's a ANOVA statistical analysis or not.
So initially for the AIM-1, we had a lot of statistics. We used a t-test, and specifically for the flasks, I believe that was definitely less than 0.0001. But when we used qPCR on that one point when there were 2 asterisks, that was closer to the probability was equal to 0.006, closer to that. The results are really more new for AIM-2, so I would have to look back into the results to see the exact numbers for the iron, where there's also statistical significance. But I know that when there are 3 stars and not 4, that would mean that the value was greater than 0.0001.
So, I'm supposed to introduce myself, you said? So I'm Corine's tall but slightly dim-witted uncle. This was a lot for me to take in. I'm probably still back on like slide 5 or something. But the question I have is, so in the AIM-3 and what you were talking about toward the very last part of the briefing, you were talking about glucose and iron in the environment, right? And what I'm trying to grasp is, you pick iron or you pick glucose because it's going to be a nutrient for these particular organisms. But you picked iron because iron is present during menses, and you wanted to see, was that the most dynamic environment to see these particular organisms do what they do in? Or was there another particular molecule that you can actually pick in order to see what you needed to see? So in other words, I know that glucose was the nutrient, and I'm having trouble figuring out or understanding the role that iron particularly played in it.
Right, that's a great question. I'm glad you asked that. So from this pathway, you can see that both species are able to metabolize glucose. You can see at the top. And glucose is a carbon source. All species would need a carbon source. And sometimes when we did some of the spent medium experiments, sometimes glucose could be depleted and it wouldn't be there. So in order to kind of negate that as a possibility for no growth, we just went ahead and added glucose, even for AIM-3. But in the case of iron, I'm glad you asked that because it could be explained more. Now I'm going to make an observation of this one graph, but just please note that this does not apply to every woman. It is only in this case, and I did choose this because it demonstrates a point that I wanted to make, but it is not the same for every woman. But if you are to look at this graph, you'll see that every time that there's a spike, this is when a woman had her menstrual cycle. And when you have a menstrual cycle, there is an increase in iron. And so this, again, was not similar. I don't want to cherry pick, but there have been other researchers have shown that when there is increase in blood or is an iron-containing source, that iron tends to thrive. Another interesting study, it wasn't published, but it was written about, showed that Lactobacillus inners did grow in MRS, which it doesn't grow without the addition of blood. And it was when 1 to 10% human blood was added and 1 to 10 of sheep blood was added. And then, but however, when they just added iron by themselves, they didn't see that growth. So a lot of people do say that a source of iron could potentially grow. Also with serum, when you add serum, it contains iron among other things. But what is it that makes them grow? We don't really know. So that was kind of why I added iron, just as a hypothesis. Maybe it could help it grow. And another thing is that MNC media, because it contains the serum, which has some traces of iron, enough for it to grow, we weren't able to remove it. And so we chose iron because it was easier to work with. We didn't have a chelator for a carbon source, so that's why we would have chosen a carbon source, and I believe Kanetta and Larry's and Professor Forney's lab tried to do a competition experiment with a carbon source. But for our purposes, we chose iron because we were able to change the availability of it.
Right, and it was present during Minzy's, like your spike show, and so
yeah. So in our experiment, it was basically using synthetic laboratory media, but this is an example of what happened. This is a different study, but this happened within the woman. This represents her microbiome. You're welcome. One more, Scott.
I have kind of a related question, maybe. So I thought it was really interesting that the two different species, one grew in CBF, the other one didn't at all. And you speculated that maybe there was something missing or the nutrients weren't there. I'm wondering if you have any ideas on what might be the thing missing, and also is it possible that there's something like inhibitory that comes from the fluid itself?
That's a good question. Thank you for asking that, Scott. So the first question was, wait, remind me the first one again, just with one word, keyword.
Yeah, whether you know what nutrients might be.
Right. So mucin. Mucin is in cervical vaginal mucus, but that's what species are known to bind to, adhere to, and some species can metabolize it. I believe like L. inners can, or L. crispidus can, and that was removed. So that could have been one nutrient that was not there specifically. And then your second question.
About maybe whether there's something toxic.
Yeah, so I would say I don't think that there's anything that was toxic because, well, maybe it's pH dependent. But from this graph, L. crispidus didn't if it was toxic, it would have decreased. And that is what happened for L. inners at pH 4. But when the pH was at 7, it didn't decrease. So I think that the results did show that we didn't really see an interference because L. inners was able to grow in L. crispidus, dominated CBS. And I think that shows that there were no molecules present or active that were able to inhibit L. inners.
Well, I have to say that this is one of the most intensive audience I've had about all the defenses I've experienced. So I'm sure you have more questions, but for the purpose of this process, we actually do have to get you guys out. And then we'll continue this defense process. But thanks everyone for coming.
take care.